Hello, welcome back. This is part 2, and let's begin. So the results were that only 2 flies were killed during the whole experiment out of the whole 1,195 houseflies used. And it was the inoculation of H7N1 at temperature 35 degrees Celsius after an incubation period of 24 hours. The researchers found out that the isolation of infected virus strains really depended on the temperature the houseflies were stored in, the length of incubation period the houseflies were in, and the concentration of virus that they were fed. Overall, though, the experiment showed a decline of virus at all concentrations and temperatures over time. It seems the persistence of the low pathogenic avian influenza strains in the houseflies alimentary tract really depended on the virus strain used, either H5N7 or H7N1. The possible variables that could have affected the amount of virus particles obtained from the houseflies could be number 1, thermal inactivation, which was a process used by the researchers to in order to kind of not allow outside particles to be included in the sample obtained. They used heat, but that could have also destroyed some of the virus particles that were being obtained from the inside the body. 2 could be the own houseflies digestive enzymes that could have just destroyed the virus particles present in their digestive system. 3, normal other housefly processes such as regurgitation and defecation, we could have just expelled virus particles before the harvesting happened. The importance of this, though, is that the persistence of low pathogenic avian influenza virus in houseflies could result in higher probability of mutation into higher pathogenic avian influenza virus strains. Also, the longer persist in the system of the houseflies, there's a higher chance it is able to be transmitted to another fly or maybe another animal. This is really vital to poultry farming, as these are animals that flies tend to swarm and pester, and ingestion of infected flies by chickens or even similar birds could result in AIV infection, depending on the strain and amount of infected flies. It would have the most devastating effects on close-knit poultry farms, which are the ones we're normally used to, where chickens are so close to each other that if one got infected, the transmission would be way faster, right? It would occur really quickly. And to recap, this study showed that avian influenza low pathogenic strains can be isolated after 1 day of ingestion from the housefly's alimentary system. Although factors such as temperature, incubation period, and concentration of the virus strain affects the persistence of virus in the houseflies. The danger is that the low pathogenic and high pathogenic strains of avian influenza virus can have mild to serious health effects, and the possibility of low pathogenic avian influenza virus mutating into high pathogenic avian influenza virus can have drastic effects on poultry farming and human health. So what is next? More research is needed to determine if infected flies even have the potency to infect bigger animals such as humans. Just because it's an infected doesn't mean it can pass it on, right? And that's something we need to look into it more. Thank you very much.